Hello students, this is Dr. M. Bhushanam, Assistant Professor in Zoology, Maharani Science College for Women, Bengaluru. Hope all is well. Take care of your good health during this pandemic time. In today's session, we shall discuss on the embryological evidences of organic evolution. The objective of today's study is to study the embryological evidences related to organic evolution. The content includes, under the evidences of organic evolution, the type embryological evidences. Well students, if you look at the slide picture, you find 5 embryos, isn't it? All the 5 embryos look alike. They look similar with the features, right? But to your surprise, all these embryos do not belong to the same animal. It clearly indicates here some stage of embryonic development is related to the first row of picture. The down row indicates the organisms of them. When we look at here, it very clearly indicates that at some stage of development of embryos, it looks similar to each other, indicating that they are evolved from a common ancestor. You can see here in the picture, the first towards the left side indicates the embryo of the fish. Similarly, the second one is of salamander, third one of tortoise, fourth one of a bird, chicken, and fifth one the human beings, the man. According to this slide picture, it resembles same at embryonic stage that we see in the first row, and it is an indication that we are all related. This is the importance of embryological evidence of organic evolution. It means that otherwise, the embryonic developmental aspects of the organisms are looking similar to each other at one stage in their development. But when we look at the final stage, that is the adult stage of these organisms, it totally differs. That is what you find in the slide, right? In the first row, you find the embryos of different organisms. In the second row, you find the organisms related to them, the adult organisms or the fetal organisms. Never there is resemblance in the second row, but in the upper row, we find the resemblances. This is what we call it as embryological evidences of organic evolution. As we all know, evolution refers to the study of adaptations and changes in the organisms over a period of time. It is due to the changing environments that the organisms faces. Evolution results in the formation of a new species of organisms, which we generally call it as speciation. Sometimes the organisms that fails to adapt to the newer environment will die, and such organisms will become extinct in near future. So down is the picture, which is very popular example of the adaptive changes that an organism possesses to develop a new character in them. You can see series of the changes in the neck part of the giraffes, and the shorter neck giraffes that lived as ancient organisms now have come to a stage where you find the tall or long-necked giraffes or tall giraffes. The second part, first part is evolution. The second part is embryology. Embryology is a study and analysis of embryos, the developing embryos and the developmental stages of different organisms. Most vertebrates shows similarities with the embryonic stages. It indicates that they are all evolved from the common ancestor. This science of embryology thus acts as an evidence for the process of organic evolution. We also call it as the Darwinian ideas. So down is the picture that represents the different developmental stages of human beings. So all these stages together we call it as the study and for different developmental stages of an organism that comes under the concept of embryology. As we all know, different organisms of different classes developing from their embryos shows much of similarities as we see in the first row in the diagram. But the embryo of different classes of organisms do show the similarities at some stage of development. And their adults, when we look at, they look different or they appear different to each other. This kind of examples will suit much for the evidences of embryology to support the concept of organic evolution. As in the picture down, as we have discussed earlier, the first row indicates the embryos of different classes of organisms, shows the similarities, whereas in their adult stage they look much different. So this is what is the through light related to the concept of embryological evidence. Embryos of chicken or cat and human beings, they look similar at most early developmental stages. Scientists have identified nearly 60% of these proteins coding genes remain common or same between these stages of the organisms. So the organism that shows the similarities in different stages in such embryos, the proteins related to the stage when they have isolated, the coding genes were same and the proteins what they formed was also same of 60%. And it is an indication that they are all originated from a common ancestor. So down in the picture when we look at, towards the left side is the human embryo of 32 days, the cat embryo, the second one, which is in black and white, whereas the colored ones when we look at, the first one is the chicken embryo, the second one is the human embryo. They all look similar, indicating that most of the proteins that we have remains common between the embryos of different species of organisms. This concept became an interesting field and developed a new branch of science called as evolutionary developmental biology, also referred as evolutionary embryology. In short, it is called evo-devo. Evo stands for evolution, devo stands for developmental biology. This branch of science originated in the 19th century by scientist Alexander Kowalevsky, because he used the embryonic stages of development to classify the organisms. Down in the picture we can see Alexander Kowalevsky, and towards the right side is the picture of the frog's tadpole and Ascidian tadpole. Both look similar, but there is certain changes in their adult stage. So the embryonic stages were used of different organisms to classify the organisms. So this was done by Alexander Kowalevsky. For example here, initially the sea squids, also called as tunicates, were placed under phylum Mollusca. The reason was the adult showed a very soft body like that of a mollusk. But it was this Alexander Kowalevsky who studied its larval stage, where he could find the structure of a notochord, structure of a neural tube in the larval stage. So he placed the organisms of sea squids under the group of cardates. So at present we follow the position of tunicate is seen under cardates. Also the present DNA analysis of these tunicates have shown that the concept given by Kowalevsky was correct. Down is a picture where you can find the tadpole larva of Ascidia and the adult organism of Ascidia. Nothing but the sea squid there, okay? So in this lane, German scientist by name Ernst Haeckel put forth his ideas in the form of a theory called biogenetic law or recapitulation theory. It's a popular theory under evolutionary biology. This theory states that ontogeny repeats phylogeny. Students, please remember this concept. Ontogeny repeats phylogeny. What does this particular concept say about? Ontogeny means developmental stages of an organism. Phylogeny means the evolutionary history or the ancestral history of the organism. So during development stage of an organism, the organism will repeat the features of their ancestors in the embryonic development. So some stages of the embryo appear to be like that of their ancestor. That's what the concept means here. Based on this concept, Haeckel gave the drawings of early embryos of different organisms which looked similar and shared the common features in them. So recapitulation concept here means repetition. So embryos will repeat the evolutionary features of ancestral characters of the organisms during their developmental stages. So down is the picture that represents in the first row, top row, the embryos of that organism. In the second row, slight modification bringing them the distinctive characters in the organisms. In the third row down, it represents the organisms of the adults, which are totally different, whereas the first row at the top indicates that the larvae are all, the embryos are all from the same group of organisms. So that is the ancestor of them will remain common or same. So the evo-devo concept, that is the evolutionary developmental biology concept, is based on the similarities or homologies of embryos of different organisms. So different embryos that shares the common characters are the basis for the evolutionary embryology. There are a list of homologies of embryos which are as follows. The first one is homology in early embryonic development. The second one is homology in the late embryonic development. Third one is recapitulation process. The fourth one is retrogressive metamorphosis, and the fifth one is neoteny. So based on these 5 important concepts, we are going to understand how embryological evidences will favor the process of the organic evolution. Otherwise, how the embryology gives the evidences or proofs to say that the organic evolution has taken place on the earth. Let's now understand these homologies of embryos at different stages one by one. The first one to start is homology in early embryonic development. Students, remember here, early embryonic development, it starts with the stage of zygote to gastrula predominantly. We know that all multicellular organisms from poriferans to mammals, when we look at it exhibit a common pattern of developments, such as the starting stage is the zygote, and the zygote is an unicellular fertilized egg cell stage. So down in the picture, when we look at the fertilized egg, is a diploid stage from which the rest of developmental stages will start. So this fertilized egg or the zygote stage remains common stage in almost all the multicellular organisms, indicating that all multicellular organisms have originated from a common ancestor indicating the process of organic evolution. Similarly, all multicellular organisms show the zygote undergoing mitotic divisions called as cleavage. So we have latitudinal cleavage, meridional cleavage, bringing in the multicellular nature for the embryo. So this process of mitotic divisions, we call it as cleavage, which is common in all the multicellular organisms. So from a unicellular stage, we get a multicellular stage of organisms through the process of the mitotic divisions of cleavage that forms a common stage among the multicellular organisms. Also, the stage of cleavage results in a multicellular stage, that is unicellular stage of zygote is now turned to be the multicellular stage, which we call it as morula. Morula refers to mass of cells here. This morula stage of developing embryo will now show a central cavity at this mass of cells called blastocoel, with a layer of outer cells or cell cover we call it as blastoderm. So this blastocoel is not an empty cavity, it is filled with the albuminous fluid to form the coeloblastula stage, and we also call this stage as Blastula stage, and the process is referred as a Blastulation process. So down in the picture, after morula, we find a mass of cells enclosing a space filled with the albuminous fluid called as the blastula or coeloblastula. Now, so till blastula, we have seen almost all organisms exhibiting the common feature. The next stage, where the blastomere cells of blastula start showing this special kind of cellular movements, which we call it as morphogenetic movements, like epiboly, emboly, invagination, involution, convergence, divergence, etc. So these morphogenetic movements within the blastomeres or blastomere cells will now result in the formation of a newer cavity called Archenteron or Gastrula with two definite layers for the embryo. And this stage where Archenteron or Gastrula is formed along with two definite layers, we call it as Gastrula stage, where the outer layer is called ectoderm, the inner layer is called endoderm. Students, remember, this endoderm layer will form the lining of the new cavity, which we call it as gastrocele. So lining of gastrocele is called as endoderm, whereas outer layer we call it as ectoderm. So the opening of the Archenteron cavity, which is enclosed within the endoderm, is called as blastopore. Students, remember, this two-layered stage is called as diploblastic condition of the Gastrula stage, which is commonly seen in cnidarians and the Poriferans. In most other organisms, we find three germ layers which are formed from the Gastrula stage itself, and those organisms are called as triploblastic condition. It is common from the group of organisms of platyhelminthes to mammals. So remember, in diploblastic condition we find two layers of two germ layers, that is outer ectoderm and inner endoderm, whereas the triploblastic condition includes three germ layers, namely outer ectoderm, middle mesoderm and inner endoderm. Now, the ectoderm is the outer layer of the embryo that forms the epidermis, and this epidermis will differentiate to form the skin and the nervous system when the organism develops into adult. Whereas the inner endoderm, it differentiates to form digestive system, then the respiratory system, especially the alveolar cells, lungs, etc. develop from this endoderm. Whereas the middle mesoderm forms the rest of the body systems like circulatory system, excretory system, reproductive system, then muscular system, skeletal system, dermis, etc. Students, remember, this is the stage of triploblastic condition, which is common in the organisms of phylum Platyhelminthes to the chordate group. Now, the next condition called coelom differentiation. It is also a common process that we see in most group of organisms. As we have seen, zygote stage is common in almost all organisms. Multicellular stage called morula, cleavage, then blastula stage, gastrula stage, germ layers of triploblastic, diploblastic condition, etc. They all remain common in almost all organisms. In the same lane, coelom differentiation also remains common between the organisms. Example, acoelomate condition is restricted to the organisms of poriferans to platyhelminthes or flatworms, where coelom is absent in the organism. Then pseudocoelom is seen in the organisms of aschelminthes. But now an important condition called eucoelom is seen commonly from the organisms of annelids to mammals, indicating that we are all originated from a common ancestor. Down in the picture when we look at, the first one is acoelomate condition. So you don't have a cavity between the endoderm, mesoderm and ectoderm. Whereas between endoderm and mesoderm there is a cavity seen in the second group called pseudocoelom, but it is filled with the fluid matter. But a definite cavity is formed in the eucoelomate organisms, indicating that the eucoelomates, that includes annelids to mammals, are formed from a common ancestor. So, when we look at the early embryonic stages in the point 1, where homologies of early embryonic developmental stages such as zygote stage, cleavage stage, morula stage, blastula stage, gastrula stage, then coelom condition, germ layer condition, etc., they all remain common in most groups of organisms, indicating that all these organisms have originated from a common ancestor. So, this process access the embryological evidences of the concept of organic evolution. Now, the second type is similarities or homologies in the late embryonic developmental stages of vertebrate organisms. It includes the characters like presence of gill clefts, notochord, tail, rudiments of eyes and ears, etc. In the picture, when we look at the violet color shade indicates the gill slits, which are seen for the fish embryo, reptilian embryo, bird embryo, and human embryo. Because all these embryos will be enclosing or are surrounded by the fluid matter. This fluid matter, or amniotic fluid as we say in the higher vertebrates, the organism need to breathe. So this gill slits helps them to have this breathing process. And you also can find a tail in all these organisms. So presence of tail, presence of gill clefts are common in the embryos here. The presence of notochord is another basic character of almost all the vertebrate organisms. It appears at different positions in different chordates. For example, here in cephalochordates, it is present between anterior to the posterior ends of the body. Whereas in another group called urochordates, it is restricted to only the tail portion, that is half the portion of their body. Whereas in euchordates, it's modified to form the vertebral column. The picture down when we look at in Ascidian, the example for the urochordate, the adults lack notochord. So notochord is absent in the adult organisms of this urochordate. But it is present in their larval form at the tail location. That's what you see here. Notochord is present in the tail region. So you find notochord not in the adult but in their larval stage, where related to the tail portion alone. Similarly, the limb buds or rudimentary limb buds, they are seen in the embryos of all tetrapods. Tetrapods includes the organisms that shows presence of two forelimbs and two hind limbs, a pair of forelimbs and a pair of hind limbs. It includes examples of amphibians to mammals. There are the organs, the limb buds are the structures which forms the organs of forelimbs and hind limbs in future. It is same or its location remains same in the embryos of different class of the tetrapods. It is same with the features of eye and ear rudiments also. So the late embryonic stages that shows presence of gill clefts, notochord, limb buds, then ear and eye rudiments, they are all commonly present in the late embryos of all different classes of tetrapods, indicating that they are all originated from a common ancestor. This is what is the ideology. So this forms a strong embryological evidence of the organic evolution further. The next homology concept is the concept of recapitulation theory, which is also called as biogenetic law of Ernst Haeckel. It is one of the most popular theory of embryological evidences of organic evolution, which was given by Ernst Haeckel in 1860s. This theory states that ontogeny repeats phylogeny. In short, to remember, we call it as ORP. O stands for ontogeny, R stands for repeats or recapitulation, P stands for phylogeny. So, ontogeny repeats phylogeny or ORP concept here. It means that during embryo formation of an animal, from fertilization stage to the late embryo, most similar stages are seen in the organisms, or the organism will repeat the features of their ancestors. So this is the specialty related to the recapitulation theory of Haeckel. So down is a picture that you see where the first row at the top are the diagrams drawn by Ernst Haeckel of different embryos of different class of organisms like fish, salamander, turtle, chicken, rabbit and human beings. This biogenetic law is also known as Meckel-Serres law because it is given by Etienne Serres and Johann Friedrich Meckel. This Meckel suggested the term that we make use in the law called recapitulation. So this concept of recapitulation was given by these two scientists. Meaning of the recapitulation is repetition. In the last statement, we find ontogeny recapitulates phylogeny or ontogeny repeats phylogeny. So this law is also called as law of correspondence as it relates to evolutionary concept with that of developmental sequences. So during the developmental stages of an organism, the ancestral characters gets repeated in them. So that's what the biogenetic law of Haeckel predominantly talks about. The law predominantly makes use of two words. Number one, ontogeny. Number two, phylogeny. So what does it mainly indicates? It indicates that during the developmental stages, the ancestral characters gets repeated in the embryos. We have two words in the concept. Number one, ontogeny. Number two, phylogeny. Ontogeny refers to various developmental stages from the zygote till the formation of a mature individual or an adult. In short, it simply refers to the developmental history. So when we look at the picture here, the developmental stages of different stages of the human beings we find. So something like this, every organism will have their developmental stages, which we call it as ontogeny. The word phylogeny refers here to the concept of evolutionary history of an organism. So what exactly evolutionary history refers to? It implies to the relationship between various organisms based on the concept of genetical and physical features. So genetically, physically, the features of the organisms need to be present. So here it is represented with the concept of phylogenetic tree. It starts with a common ancestor. Phylogenetic tree always starts with a common ancestor. It starts from that common ancestor and it shows many number of branches showing the similarities with the genetical and physical features. Like this, the growth of this evolutionary tree is seen. So it is done by using the intensity of close relationship between the organisms. Then the branchings also are shown with the help of the fossil study available or sometimes even the hypothetical assumptions that we make use. So that way phylogeny is concentrated. For example, here the fertilized egg cell stage called a zygote can be compared to a single celled stage of diploid organisms or diploid animals like protozoans. So zygote cell is the ancestor of protozoans. Later to zygote stage in the development we find cleavage, morula, blastula, indicating the multicellular nature. So they are the Blastula stage is considered as the ancestor for the colonial protozoans, the multicellular metazoans. The next stage to Blastula is the Gastrula stage, where we find diploblastic and triploblastic condition. So Gastrula is considered as the ancestor form of triploblastic and triploblastic organisms. That's what you find in the picture down. So you find phylogeny, ontogeny repeats the phylogeny. Example is given here. Ontogeny, where developmental stages you find. One is the X stage, the second is Blastula, third is Gastrula, fourth is tadpole, fifth one is adult frog. So that is the developmental stages or ontogeny related to frog. Phylogeny of it, when we look at X stage, can be the repetition of protozoans. Blastula, poriferans, Gastrula, coelenterate, tadpole appears to be like that of a fish, and adult frog is an amphibian. So during the developmental stages, the developmental process will show the ancestral characters in the organisms. This is what we call it as the concept of biogenetic law, where ontogeny repeats phylogeny concept. So just now we have understood a tadpole stage of frog resembles a fish almost in having structures like fins, gills, latoline sense systems, etc. So ontogeny of amphibians, that is developmental stages of amphibians, shows the phylogeny of fish, because the tadpoles will resemble the fish structure here that you find in the picture down. Same with the birds and whales. The embryo of birds develops the teeth, which disappears when they become adult. So it indicates a reptilian trait in the birds. So birds during their developmental stage shows their ancestors of reptilian characters in them. Similarly, fetus of whales, they develop a hairy coat. The cover of the fetus the body cover of the fetus will have a hairy coat. But when it gives birth, the newborn will show loss of hair totally. So presence of hair is a special character which is not related to the organisms here. So you know that whales, dolphins are considered to be mammals. Okay, so we show the presence of hair in the lower mammals. So that's the character gets repeated here. We also know that mammalian group of organisms do not have yolk in their eggs. But to our surprise, we find still the yolk sac formed in the embryonic stage. When yolk is there, yolk sac should be formed. When yolk is absent, why yolk sac need to be formed? There is no need. But it is present, indicating that ontogeny repeats phylogeny, as yolk sac is present in our lower group of organisms like birds. So features of hollow nerve cord, notochord, gill slits, tail, two-chambered heart, then pronephros kidney, primitive kidney it is. They are all the characters present in a human embryo, which is of earlier stage, early human embryo. These are also the features that you find in a tiny fish, where you find a gill slits, a tail, a notochord, a nerve cord, two-chambered heart, pronephros kidney. So it's an indication that the organisms repeats the ancestral characters during the developmental stages. Similarly, the 7-month human embryo resembles a baby ape with the body fully covered with the hairs and having a long forelimbs. So down in the picture when you look at, yolk sac is present in mammals, which is the character of reptiles and birds. And we do have the presence of body covered with the hairs and long forelimbs, which is a character of the chimpanzees. But we find that character in 7-month-old human embryo also, resembling we share the characters of our ancestors during our embryonic stages. Students, remember, these are all the examples which will help us to support the process of organic evolution through the concepts of embryology. There is one more law, that is law where ontogeny repeats phylogeny, is based on the law of terminal addition. So what is added here at the end? Here the embryos will develop more specialized features at the end of the embryonic development or when it enters into the adult stage. Example, amphibians are evolved from the embryo of fish. So when a fish character gets into amphibian character by adding a new stage at its end. Until then it shows the presence of a character of a fish, but by the end of the embryonic development, the characters of amphibians gets added to it. So that's what the concept of law of terminal addition refers to. So students, remember, the tadpole larva resembles the fish. So it appears to be like that of fish. Later it develops the limbs, forelimbs and hind limbs, tail is last, then it forms a tiny frog. So this stage is related to the amphibian character. So fish characters are last, amphibian characters are imbibed into the organisms or newly gets developed into the organisms. That's what the law of terminal addition talks about. There is one more law that supports ontogeny repeats phylogeny. So law of correspondence, law of truncation and law of terminal addition. These three laws supports the biogenetic law. Okay. So law of truncation, it's a concept associated with the biogenetic law of Haeckel. So it refers to the progressive stages of development get shortened to ease the life pattern of an organism. So during the developmental stages, certain of the stages gets shortened by its period of time. For example, gestation time from a long developmental duration. Down is the picture when you look at different gestation period, we find different structures getting developed. By the end of the 9th month, you find a completely formed embryo develops into a fetus. So completely developed fetus you find at the end of the 9th month of developing stages of human beings. So the gestation period will not extend to a year or 2 or 3 years, etc. Why? Because the shortening of gestation time will help the organism to ease their life pattern. So that's what is the importance of a law of truncation. The next important homological feature of embryological evidence of organic evolution is retrogressive metamorphosis. So retrogressive metamorphosis refers to the larva of an organism exhibiting the basic chordate characters, but their adult forms loses them, they don't show. Example here is the Ascidian. So in Ascidian tadpole, the larval form is a free swimming form showing all the basic 3 chordate characters. Presence of notochord, gill slits, etc. They all totally gets degenerated by the time it enters into adults. During the development, the metamorphosis process shows the larval stages are entirely different from that of the adult stage, because during the process of development, larval stages shows the advanced characters in them than their adults. So notochord, nerve cord is the advanced character of a chordate, but it is present during their larval stage, not in their adult. So when it goes, metamorphosis goes in a reverse process, we call it as retrogressive metamorphosis, where larvae will be showing advanced characters of a chordate, but not their adults. Adults will show degenerating characters because they don't show presence of notochord, nerve cord, etc., which we call it as retrogressive metamorphosis. The next homological feature in support of embryological evidence of organic evolution is neoteny. The word neoteny is derived from a German word neoteny, and it was given by Coleman, according to which the term neoteny refers to juveniles exhibiting adult traits. So juveniles are referring to the larval stages. They exhibit the characters of their adults. They behave like adults. When you look at here the concept of axolotl larva of ambystoma, the larva fails to undergo metamorphosis due to environmental conditions again. The larva will show presence of development of gonads, and they attain the sexual maturity in their larval stage itself. So larva will develop gonads and attain to the sexual maturity. This is what we call it as neoteny or pedogenesis. So what's happening here? The larval characters are retained in them. Along with it, it will show the adult sexual characters also. So indicating that the larval forms showing the adult characters in them are undergoing sexual maturity in them is also an embryological evidence of organic evolution. So down is the picture where you have at the top the ambystoma adult, and down is the larval form, which we call it as axolotl larva, where we find specifically the concept of neoteny. The next popular concept of embryological evidence of organic evolution is von Bayer's principle. We also call it as von Bayer's law. Students, remember, it is a very important concept, most accepted, like that of Haeckel's law. So here the concept of von Bayer's principle is based on 4 major postulates. It is also very important for your main exams. The first postulate is the general features of large group of animals appear first. The common characters that we find between the classes of different organisms starts appearing first during the development, then follows the specialized characters in them. For example, here when we look at all developing vertebrates will appear almost similar features till the stage of gastrulation. But after the gastrulation, their cell determination will take place, then cell differentiation will take place, and the different germ layers will start developing the organs of their own. Okay, so that way we find differentiation in the organism. But in the first stage, common characters related to all the organisms will start developing. This is what the first postulate talks about. So down is the picture when we look at the first row. It is related to the first postulate. Excuse me. The first postulate says all the general features starts developing in the first stage of development. So later special characters starts developing into the organisms. So it is only later, the first phase where the general characters are formed, the special characters are followed. So it goes in an order, class, order, etc. It goes that way. So finally a new embryo will emerge showing special characters in them. So all vertebrates will have gill arches common, notochords common. spinal cords common, primitive kidneys common, nervous system common, etc. So these are all primitive characters which starts appearing in the initial stage, which later follows the developmental stages related to special characters. For example, if you take the fish, fins are present, it's a special character related to them. Then when you look at the tortoise, they develop the carapace, plastron. That is a special character, it gets formed in later stage. Feathers in birds, it gets formed in the later stage. So likewise, we find special characters are formed in the later stage. In the first stage, all the common characters gets formed in the organisms. The second postulate is less general characters are developed from more general characters until finally the specialized characters appears. What does this concept talks about? We know that all vertebrates initially will have the same type of skin. So every organisms, right from the fish to the mammals, we all will have the skin, primitive skin. Later, the skin develops scales in case of fishes, reptilian scales in case of reptiles, feathers in case of birds, hairs, claws, nails in case of mammals. So most special characters gets formed after the formation of the general characters. How? Less general characters are developed from the more general characters. Then finally specialized character appears in them. So similarly, the development of limb, especially in case of tetrapods, it remains same. But later, they show the difference of limbs to form the legs, then wings, then arms, which are differently appears in different classes of organisms. But the limb structure, the limb birth will remain common in all the organisms, as you find in the picture down here. The next postulate is the embryo of given species, instead of passing through the adult stage of their animals, departs more and more of them. Embryo of a given species, instead of passing through their adult stage of lower animals, departs more and more from them. What does it mean? During the developmental stages of organisms, instead of, that ontogeny repeats phylogeny, where the ancestral characters get seen in the developing embryo. What kind of ancestral characters are seen? It is not the adult ancestral characters, it is their embryos. Ancestral embryonic characters are seen, or the organisms shows the ancestral embryonic characters, not the adult characters. So for example, the visceral clefts of embryonic birds and mammals do not resemble the gill slits of adult fish in detail. So we do have gill slits like that of fish, but the gill slits what the embryos of the birds and mammals show, it is not like that of the adult fish. Instead, it is the embryo of those fishes what the gill slits it shows, that similar gill slits are shown by the embryos of the birds and mammals. So remember, the adult characters of our ancestors are not repeated in us. Instead, the embryonic characters gets repeated. This is what the third important postulate concentrates about. So fish will preserve and elaborate these gill slits to form the gill structures and gill slit structures, whereas in case of mammals, that the gill slits gets modified to form the eustachian tube. Gill slits are nothing but the communication between, the opening between the pharynx and the external environment. So that space is brought about by the eustachian tubes, where the mouth and the ears are getting connected. So gill slits are modified to form eustachian tubes in case of we the human beings, whereas gill slits will remain as gill slits in case of the shark, that is fish embryo. The next important feature is that all the early embryo of higher animal is never like a lower animal, but like its early embryo. Again, a concept which indicates that embryos of the organisms that shows the phylogenetic repetition in them will show the embryonic characters of their ancestors only. For example, human embryos will share the characters like that of embryos of fish and amphibians, reptiles, birds, etc. So, embryos of these organisms of lower group may be repeated with the characters in case of human embryos also. So, later the mammalian embryos will diverge to form different stages of development and starts developing a newer characters in them. So, this is what the importance of Van Bayer's principle talks about. So, Van Bayer concept shows that common pattern of development is commonly seen in all the vertebrate development. So, most common characters are formed first, then follows the special characters. So, this is the speciality related to Van Bayer principle. So, germ layers give rise to different organs, and this derivation of organs is constant between the organisms of fish, frog, chick or mammals, etc. But remember, even though they develop from the common germ layers, the structures doesn't remain same. The meat of us, the flesh of us is not similar to that of flesh of a fish or a frog or a chicken. Reason is that the differentiation process is a specialized character that we find to different organs in us. So, this is what Van Bayer's principle talks about. Well, students, with this we are completing the concept of embryological evidences of organic evolution. Few MCQs to understand. First one, the recapitulation theory was given by, who's the scientist? It is A. Ernst Haeckel. Second question, Haeckel's theory of recapitulation means that all organisms start as an egg, progeny resembles their parents, life history of an organism reflects the evolutionary history. The last concept is last body parts are regenerated. So, answer is C, because life history of an organism reflects the evolutionary history, nothing but the recapitulation theory of Haeckel concept. Ontogeny repeats phylogeny. Answer is C. Appearance of teeth in the embryo of bird proves what? It proves ontogeny repeats phylogeny, yes. Evolution has taken place, yes. Birds have evolved from reptiles, yes, because the tooth is the indication where reptilian character gets seen in the bird. Fourth is all the above. So, D is the correct answer, all the above. Question 4. Who observed that embryos never pass through the adult stage of their ancestors? So, among these scientists, who have observed that embryos will never pass the adult characters in them? It is A. Van Baer, because Van Baer principle says that adult characters are not seen in the embryonic stage of development. Instead, embryonic stages of our ancestors are seen in us. Alveolar cells of lungs arise from which of the germ layers? It is endoderm. So, answer is B. The presence of gill slits in the embryos of vertebrates supports the theory of what? So, presence of gill slits in the embryo of vertebrates is a recapitulation theory. So, answer is D. Which of the following represents the correct order of evolution? So, from lower to complex forms it should be amoeba, cycon, hydra, ascaris. That should be correct. Cycon, hydra, amoeba, no. So, amoeba is given rise to cycon. Amoeba is protozoan, cycon is a porifera, hydra is a coelenterate, ascaris is a nematode. So, the order is correct. Answer should be A. Yes. Even after attaining sexual maturity, larval characters are retained in them. The organism has got sexual maturity, but still the larval characters are seen in it. Otherwise, larvae will show sexual matured characters in them. What is it? It is B. Neoteny. In axolotl larva, what we find. Then, retrogressive metamorphosis is peculiar to which of the groups? It's urochordate where ascidian tadpole is seen. So, answer is A. The last one. The general features of a large group of animals appear earlier in the development than the special features of smaller group. What does it refer? It refers to Van Bayer's law because the Van Bayer law itself states in the first postulate the same. So answer is D. Well, students, what's the outcome that we have studied? We have understood the role of embryological evidences in the organic evolution. The references includes Wikipedia and Britannica.com as web references. The book references includes Organic Evolution by Veerabala Rastogi, Evolutionary Biology by Veerabala Rastogi, and Cell Biology, Genetics, Molecular Biology, Evolution and Ecology by Verma and Agarwal. Kindly post your comments and queries to my personal WhatsApp number. Thank you all students. Take care of your good health during this pandemic time.